The overall goal of this procedure is to study age-dependent genomic instability in yeast by combining the chronological lifespan with a set of simple DNA damage and mutation assays. This is accomplished by first sampling chronological aging cultures every 2 days and checking for viability by colony-forming unit or CFU formation. The next step of the procedure is to examine the DNA mutations by sampling aging cultures and plating cells on mutation-specific selection medium. Ultimately, results can be obtained that show changes in DNA mutations during yeast chronological aging through normalizing the mutation assay results to the viability.
This method can help answer key questions in the aging field, such as the mechanisms that link aging and genomic instability in higher eukaryotes.
To assay for yeast chronological lifespan, or CLS, first inoculate a single yeast colony into 1 milliliter of synthetic complete glucose, or SDC medium, and incubate overnight with shaking at 30 degrees Celsius. On the following day, dilute the overnight culture into 10 milliliters of fresh SDC medium to an OD600 of 0.05 to 0.1. Incubate with shaking at 30 degrees Celsius for 3 days. On day 3, when cells are in stationary phase, dilute each aliquot 10,000 times in autoclaved water. Then, remove 2 10 microliter aliquots from the flask and plate 10 microliters of each diluted culture on a YEPD plate. Incubate the plate at 30 degrees Celsius until colonies arise. The number of colonies arising, or colony-forming units, from the day 3 culture is considered 100% survival. Continue removing aliquots from the aging culture in subsequent